Hey, welcome to our first unit summary here, and this is going to be just a quick run through of everything that we are going to see in this unit. Our first unit's theme is emergent properties, and emergent properties is something which is totally fascinating because basically it's when you take very small things and put them together and join them together, they become bigger things, and those bigger things have property that neither one of the smaller things had prior to it. Alright, so when we look at biology, which is a study of life, we realize that life itself is an emergent property. So all those things that you studied in chemistry and in physics are going to be the result, are going to allow living things to take advantage of combining those characteristic properties that you learned in chemistry and physics, and the property that's going to emerge is something that we refer to as a phenomenon called life. Right? Excuse me. And now if we look at this, it's a pretty easy question for you here, but which of these following molecules is alive? And it's probably something not molecules, but which of the following things here that you see in this list is alive? And it's very likely that you'll be able to find out that only one of these is alive, and that is, of course, cells. So we have atoms make up molecules, and then viruses are made up of many, many different kinds of molecules, but literally the viruses themselves are not alive. They don't fit the definition of life, and so they do not, or they lack that emergent property. Cells, which are combinations of other kinds of molecules, they are alive. And so life itself emerges from the interactions of these other molecules and in different combinations. Alright, so a quick review of chemistry for you. We're assuming that you already know about atomic theory, so that if I drew this little molecule for you here, I'll blow it up and bring it to the front for you. And we look at the molecule, you know the structure of an atom. And so you understand the nucleus and the protons and neutrons found in there, and you found that the electrons are orbiting around the nucleus, and that the protons determine the element, and the mass of the atom is based on what you see in the nucleus. The electrons play another role. Their distribution around the nucleus is going to help describe how they behave towards other atoms. And so I'm assuming that you would know those kinds of things. So a little bit about atomic structure. Now we are going to pay attention to valence electrons, and hopefully you remember valence electrons. Pull that baby out just to remind you. Are the electrons that are going to be on the orbit that's farthest away from the nucleus, and these are the ones that are going to interact or not interact with the atoms that are they're coming in contact with, other atoms they're coming in contact with. Well, along with valence, then we have this other property that we're going to be paying attention to. It's called electronegativity, and this is also a characteristic property of atoms. And electronegativity was discovered by Linus Pauling, and it moving from left to right is increasing electronegativity, and the atoms that have the highest electronegativity would be fluorine, oxygen, are going to be way up there near the top, and they're going to be in the darker blue. And electronegativity is a property of atoms. It's their attraction of one atom for the electrons, valence electrons of another atom. The higher the electronegativity, the stronger that attraction is. And then electronegativity is going to determine something that we call polarity, and polarity, hopefully you remember, is a charge difference, not like the north and south pole, but a charge difference. So you can see down here, if we have a bond between two atoms here, and this is a sodium and a chlorine, if that bond is one where the sodium is donating its atom or electron to the chlorine, then this is going to form an ionic bond, and this is going to have a permanent polarity. The sodium will be positively charged and the chlorine will be negatively charged. Now, if the atoms are the same as we see here between these two carbon atoms, then they have equal electronegativity, they have no polarity, and they are called a nonpolar covalent bond. And that lack of polarity means that the charge is equally distributed between the two and doesn't lean one way or the other. So in the case of sodium and chlorine, there's constantly a charge. In the case of carbon with carbon, there is never a charge. And then we have this one that falls in between, which is where many of our molecules exist, and that is where there is a difference in electronegativity between one atom and the other. So if you remember from here, The nitrogen, or the oxygen, is very high electronegative, 3.5 on the scale. The hydrogen is kind of moderate. It's 2.1. It's not low, but it's moderate. And so it has a weaker attraction for its own electron, its own individual electron, than the oxygen does. And so as a result of that, when the electron is going to spend more time in orbit around the oxygen, less time in orbit around the hydrogen, and that is going to lead to a charge difference. Unlike the ionic bond, it's not permanent because the electron does orbit around the hydrogen at least a little bit. You see the little gray areas there. And then it does orbit then back around the oxygen. So when it's around the oxygen, it's a negative charge, and the hydrogen has a positive charge. So permanent, never, and it goes kind of on and off. It alternates between having a charge and not having a charge. So it kind of fits in between those two in terms of its characteristic properties. So that's ionic, covalent, and then polar covalent. All right? And so we can see that distributed here as well with ionic bonds. Again, never shared, nonpolar covalent, always equal, never a charge, always a charge, and then these guys are sometimes a charge. That's what that partial charge is there. All right. pH, concentration of hydrogen ions in solution. So we have here different solutions have different pHs. Remember the pH scale is going to be increasing by a factor of 10 each time. So if we have a pH of 7, that means we have equal numbers of hydrogen ions and hydronium ions in there. And as we move down the pH scale closer towards 14, we see lots higher concentrations of hydronium ions, OH, and very few hydrogen ions. If we move up the scale closer towards 0, we are going to see very high concentrations of the hydrogen ions and lower concentrations of the hydroxide ions. So most living things exist in this space right here. We see seawater and saliva. So pH again is a concentration of H plus ions in solution, and also concentration, POH would be the concentration of the hydronium ions. Alright, some quick physics for you. First law of thermodynamics is going to be the one where it says that energy can neither be created nor destroyed, but can only change form. In the case we see up here in this picture, we see a human taking in food, and we're going to have some waste and then some food energy that the organism uses to survive. A plant does the same thing. It's going to convert that energy from one form, which is going to be solar radiation, to a chemical form that can be used obviously for itself and for anything else that eats the plant. So first law of thermodynamics, second law of thermodynamics, everything's going to move towards a state of entropy. Eventually, luckily for us, there's a large universe out there that absorbs lots of the excess heat that we have, and we can maintain our own order by creating disorder in the universe. Another good argument for not cleaning your room, which many of you might want to do when your parents are like, hey, clean your room, just tell them, hey, I'm trying to save the universe. I don't want to have any I don't want to create any additional entropy by cleaning my room. Alright, inertia, you guys know movement, and things that are in motion tend to stay in motion unless acted on by an opposing force. We're going to see that quite a bit. And then gravity, the force of pushing down on larger objects. And excuse me, that was really bad. But gravity is going to be a force that we're going to see on Earth that is going to help orient organisms. And you can see here in this little graphic here, you can have either positive gravitropism, which is movement towards a gravity, or negative gravitropism, where we're moving away from a gravitational source. And we kind of see this all the time in our seeds. That's what allows the seeds to be able to tell up, which is where the stem is going to go, from down, which is where the root is going to grow. And you can disorient them by growing seeds in zero gravity. That's kind of fun. Then the roots and the stems kind of grow flat. Energy, of course, we got these major transformations of energy. And so I'll go ahead and pull this one up. You can see that a little bit simpler. And in this case here, we have glucose being exposed to oxygen. When it's exposed to oxygen, it's going to be releasing energy in the form of both heat and light. If I burn a potato chip, that's going to happen every time I do it. It's kind of a cool activity. And then it's going to produce carbon dioxide and water, and of course the energy that we're going to get out of it. So energy is something that we can use for all of our reactions. Living things require energy. Without that energy, they're not going to be able to survive. And of course, one of the ways that we can measure the activity of those is through temperature. And so if we are looking at temperature as the average activity of the molecules in any source, and hopefully this little animation will work for us. Might be a little bit too big for it. So we'll hold on just a second. And it's not going to work for us. So, but if we have a colder temperature, the activity of the molecules is going to decrease. Of course, absolutely zero for any molecule is when it's not moving at all, and higher temperatures are when the molecules are moving faster. And so if we measure temperature, measuring the average energy of those things. And so temperature plays a huge role in living things. They all depend on a particular kind of temperature or range of temperatures to maintain homeostasis. Alright, our favorite molecule is water, and it has super cool properties that we talk about quite a bit. So one of those is surface tension. That's why this water strider can survive on the top of the water, because it's got its weight distributed and it's banking on the hydrogen bonds that are in water to be able to keep it, or allow it to move across the water and take advantage of prey that might not be able to do that. And then, of course, when water freezes, it forms these bonds that between the water molecules that form a crystal structure, and it forms ice on the tops and the edges of things first, and not in the middle. And that allows organisms to stay at or near close body, their regular body temperature, for longer periods of time. If it didn't have that property, then not a lot would live in really cold water. Alright, and probably one of our biggest things is water is a universal solvent, and we know that water is a polar molecule like we saw before, and as a result of being a polar molecule, it's going to be attracted to other polar molecules, and in this case here, the negatively charged chlorine is surrounded by the positively charged hydrogen atoms in the water molecule, and then the sodium atoms right up here are going to be surrounded by the negatively charged oxygen atoms. And so every time you stick something into water and it dissolves, that is because of the polarity of the molecule. And if you see stuff that doesn't mix with water, that means it's going to be nonpolar, like say oils are nonpolar. Alright, one of the other things that we're going to see as a result of this water is that it is going to be able to absorb lots and lots of energy without changing its temperature very much. So if you've ever lived in or wanted to visit California, beautiful along the coastline, because even though it's relatively low, closer to the equator, the temperatures are very mild, because we are absorbing a lot of that heat in the area surrounding it in the ocean. And as you get farther and farther away from there, of course, the temperatures rise rapidly because it's really hot in California. Of course, not in San Francisco or other places that are near the water. Alright, and then finally, adhesion and cohesion. So cohesion is where the water molecules stick together, and that's what forms these little droplets on here. And that's also what allows the water to move up the column in the tree. And then adhesion is where the molecules are going to be sticking to a surface. In this case, it's glass, and in this case, it's the surface of the tube that's transporting the water from the roots to the leaves. And those properties allow very tall trees to exist, because they can move water from their roots hundreds of feet up in the air through this properties of adhesion and cohesion. Alright, now the next thing we're going to be looking at here is synthesis and hydrolysis. And synthesis is an emergent property, because if we're going to take two smaller molecules and put them together, we're going to form one larger molecule. And every time we take two smaller molecules and put them together, we always form a molecule of water, and that is going to allow the polymer to form. And so every time we see that, that is synthesis. So any kind of synthesis, photosynthesis, for example, is going to be something like that. But protein synthesis, another example, water is going to be formed every time we take two smaller molecules and put one larger together. Opposite of that, and also reversible, is something called hydrolysis. If we add water, that is going to be causing one larger molecule to break down into two smaller molecules. So we have a specific example down here of synthesis, and that is if I take two glucose molecules, here are the HOH and the hydrogen that are going to be forming the water molecule, and when you combine those two together, you get maltose. So glucose is a sugar, maltose is a sugar, but maltose has properties that are different than the glucose molecules, and particular maltose is kind of a dark brown. Most of our sugars are clear white, but maltose is kind of a dark brown. If you ever had a malted milkshake or a malted milk ball specifically, that would be a great example of where you find maltose. Alright, so now if we take just many of those glucose molecules from the last page and put them together, then living things make larger poly molecules out of those polysaccharides. In this case, we can form amylose, which you guys know better as starch, and that's what seeds use to store energy so that they can grow. And then in animals, they form something called glycogen. So you can see there's some structural differences between amylose and glycogen, and that has to do with where the glucose molecules, all these little green circles, are binding to each other. And so that makes it a little bit different in shape, but its function is the same, and its storage of energy, and the molecule is the same. They're all glucose molecules. And it can also even do this, which is cellulose, and cellulose is going to be found in plants, of course. It's part of their cell wall. It's part of the fiber. So when we eat a plant, we have fiber. That's the cellulose we're seeing there. And then the cellulose is just long chains of linearly bound glucose molecules. So if you could extract the energy from cellulose, you'd find yourself with a lot of glucose, which is what cows and termites would do too. And they're going to be extracting by different means the energy from cellulose. We can't get it, but we can get it from beans. So if we had a bean or a seed of any kind, we get energy from it. We can't eat it from the fiber of the plant. So those are carbohydrates. These are lipids right here, and lipids are pretty simple in their structure. They're not truly a polysaccharide form, but they have two parts to them. They have a glycerol, which is the backbone right here, and they have 3 long-chain fatty acids. These fatty acids are primarily made of hydrocarbons, and hydrocarbons are nonpolar, and so that's what makes a lipid nonpolar. And then you can see sometimes they're straight, which is saturated, and those guys are saturated with hydrogen, and all the carbons have single bonds. And then these guys are unsaturated. Kind of see they got kinks in them, and that is because they have at least one double bond in there. Another kind of lipid that you want to know about is a phospholipid, and this is pretty cool because a typical lipid, like we see here, is going to be not going to mix with water because it's nonpolar, but a phospholipid actually has a polar head, and that makes it polar. The phosphate is polar, and then the lipid tail is not, so it's amphipathic. That means that it has one side that loves water and one side that hates water, and that determines its structure and helps form membranes. And finally, steroids. Cholesterol is the component of most of our steroids, and you guys know things like estrogen and testosterone. Those are also steroids, and those are also lipids. Proteins are made of long chains of amino acids. So we have the amino acid itself. It has the functional group amine and the functional group carboxyl, and those are joined together at a central carbon. And then it has a hydrogen down here, and then a variable group, which is called the R group, which is right down there, or right up there. And the variable part right here, you see, is different in every amino acid, and then the rest of this stuff is the same. And so what happens is a carboxyl joins an amine. Carboxyl joins an amine by synthesis, and when it does so, it takes 2 smaller amino acids and puts them together into a larger one, and this is called a peptide bond. And amino acid chains then form the polypeptide chains. Peptide is the bond between a carboxyl and an amine, and those polypeptides then join together and they can form our proteins. A typical protein you might see is collagen. That's going to make your skin kind of stretchy, and your bones, it gives a structure to the bones, so they allow you to support your weight. And hemoglobin is this is a linear one, and this one is called a globular protein, hemoglobin, and that is the one that carries oxygen and carbon dioxide, and that's it's carrying these little iron molecules in there, and then those are going to be the heme that carries the oxygen or the carbon dioxide. So that's proteins. And then, of course, nucleic acids. We have 2 types. We have DNA and we have RNA. DNA is double-stranded, RNA is single-stranded, but they are made of the same stuff. They're made of nucleotides, and nucleotides have a phosphate group, a sugar, and that's always exactly the same for each kind. And then the nitrogenous base. There are 4 different nitrogenous bases for each, RNA and DNA, where DNA has thymine right here, and then RNA has uracil, and then all of them have cytosine, adenine, and guanine. Be like 5 minutes. Okay. And then, of course, the different forms of RNA. So DNA typically has one form that you know about. We'll talk about other forms, but DNA has like one form. And then messenger RNA, ribosomal RNA, and transfer RNA are 3 different forms of RNA. So RNA has many different shapes. DNA pretty much has one, although as you come to here, we'll talk about more different kinds of it. All right, next is those phospholipids that we talked about earlier are going to form our membranes. You can kind of see the phospholipids in here in our little picture. Blow that up a little bit for you there. And then that membrane is then interspersed with these proteins that do different jobs. And the proteins and the phospholipids together make what's called the fluid mosaic model for our membrane. And that membrane that we see on cell surfaces is also the exact same membrane that we see on the organelles inside the cell. So if we talk about a ribosome has 2 membranes, and then it has stacks of membranes inside of it. The nucleus, or the mitochondria, has 2 membranes, one outer membrane and one inner membrane that's like really highly folded that you can see here. And of course you can see all the smooth ER and the rough ER, they also have membranes, they're also made of membrane. And the smooth ER is shaped differently and lacks ribosomes, whereas the rough ER has ribosomes. That's what the R stands for. That's what makes it rough. Alright, and then some other membrane bound, or some other organelles is the ribosome. And then the Golgi apparatus, and that is going to be one that's going to package proteins. Ribosomes make proteins. Golgi apparatus packages those proteins. Lysosomes are a package of proteins that go out and break down stuff. Like they can digest food that you've taken in, that your cell has taken in. And then in plants we see a vacuole. And the vacuole is where we store all the water, and that's what keeps a plant like upright. So when you haven't watered your plant, it starts to wilt. That's because these vacuoles over here, they are shrinking. They're getting rid of their water. And you fill them up, water it, dies, water it, dies, and it'll come back a couple of times, but you can't keep stressing out plants like that, and eventually if you keep watering it irregularly, the plant will die. So of these 4 right here, now which one of these is not a membrane-bound organelle? Alright, give you a second. Right, and the answer is the ribosome. The ribosome is not covered by membrane. The phospholipid bilayer is not part of it, and it is made of proteins and ribosomal RNA. Alright, so this is chlorophyll right here. This is a plant, and you can see what's going to make the plant leaf right here. If we take that leaf and we zoom in to a cross section, we can kind of see the stomata on the bottom, and we can see the top right here, and then we can see the vein in the leaf right there, and then we have all these cells inside, and each one of those cells is filled with chloroplasts. And those chloroplasts, of course, make it green, and then they are the ones that are going to capture the light energy. And then the chloroplast itself, is the outer membrane is clear, but the membranes themselves are made up of a molecule called chlorophyll, and the chlorophyll itself is green, reflects green because it absorbs the other light, and it has a phospholipid tail, and then it's got this hydrogen, carbon, nitrogen core sits on top, and this is basically going to sit on top of a membrane, just like we saw in the last image. It's going to sit on top of a membrane, and it's going to be anchored to the membrane. It's going to be capturing the light energy. And then finally, the types of light energy that it captures do vary. We have different kinds of chlorophyll, and we have they're going to be absorbing different wavelengths of light. So you can see in here the green wavelength of light, nothing really absorbs, and that's why the green is reflected. And then in terms of the activation, which ones are going to be causing photosynthesis to occur, we can see that there's a peak here in the purple and blue region, and then there's another peak here in the orange and red region. So those are the colors of light that are most likely to drive photosynthesis, because there's molecules that capture the energy from those sources. Alright, next is cell types, and these are classic cell pictures that you see. They come straight out of the textbook, and I have to tell you right now that there is not a living cell anywhere that looks like these. These are mock-ups, these are models, they do not look like living cells. No living cell has this exact combination of organelles in it. And you can kind of see here that the combinations of organelles that you see in these cells are actually going to be the thing that gives them their shape and their structure. So different kinds of organelles in cells give us different kinds of structures. And then the bacteria chromosomes over here, or bacterial cell here does not have any membrane-bound organelles. It's significantly smaller. Now I put 5 different kinds of cells here, and I'm wondering if you can figure out which one of these kinds of cells is a bacteria, So I'll give you a second there. And of course the answer to that is going to be E. This is an E. coli bacteria. Now these over here are eukaryotic cells, and I'm wondering if you can guess which one of these is a nerve cell. Alright, so give yourself a second there. So it's A, B, C, or D. And the correct answer is A is a nerve cell. Alright, and now you could probably guess this one, but which one of these is an epithelial cell? An epithelial cell is going to be something that's going to be a lining. Right? And the answer to that is D. So if you got all three of those right, then you can tell the different cell types. And the other thing you can tell is that different organelles are going to make these guys up. And these guys right here, these eukaryotic cells, have membrane-bound organelles, but the bacteria does not. The only membrane it has is the cell membrane. And then these guys are not real cells. Not real cells. Good, thank you. Last up is our nutrient cycles. Now you guys are probably very familiar with the carbon-oxygen cycle, and it's got two processes that are involved in it, and that is photosynthesis and respiration. All living things, well, most living things are going to carry out respiration. Only plants and some forms of algae are going to carry out photosynthesis, and some cyanobacteria. And then we are also going to have some decomposition that's going to occur that is also going to be adding carbon dioxide. So all of our carbon dioxide, our carbon, is stored in living things. All of our carbon is stored in living things, and then when those living things die, they're going to release it to the atmosphere, and then plants are going to take that up and use that to create more things. Nitrogen's a lot more complex. You have to spend some time looking at the nitrogen cycle. We're not going to spend time on it right now. I just wanted to introduce it to you because it is going to be a huge thing. Nitrogen cycle is going to be crucial for all kinds of living things. And same thing with the phosphorus cycle. These guys are much slower processes than the photosynthesis and respiration carbon-oxygen cycle. Phosphorus can be stored in rocks. It can be pooped out by animals and then absorbed in water. So those are going to be different things that you're going to, different cycles that you're going to want to pay attention to. And that's our end for introduction here. Thank you very much.